students, today we are going to discuss DNA replication. What is DNA replication? DNA replication is a mechanism through which from a parental DNA molecule, two daughter molecules are produced. That means from a DNA strand, two identical strands is produced. That process is called replication of DNA. It is a very important steps of cell division. In cell division, one phase is there that is called as S phase or called as synthesis phase. In synthesis phase, this replication process occurs. That means increase the copy number. And the process of DNA replication is semi-conservative. Semi-conservative process. So what is the meaning of semi-conservative? That means in DNA replication, suppose there is a double-stranded molecule. It has 1 5 to 3 dash form. Another one is 3 to 5. Two polarities are there. One is 5 to 3, another one is 3 to 5. So suppose this is parental DNA strand. So based on this parental DNA, suppose this is the polarity having 5 to 3. Another one, this is having the polarity 3 to 5. So based on this, these new DNA strands are produced. These new DNA strands are produced. That means one strand is old. Based on this strand, this again it produces the double-stranded molecule. That means that is the parental DNA strand. From this parental DNA strand, two daughter strands are produced. That is called DNA replication process. And why it is called semi-conservative? If you observe it, based on this strand, this old strand, this new strand is produced. Like that, for this double-stranded DNA molecule also, based on this strand, this new strand is produced. That's why it is called semi-conservative. It is not like that. This old strand are already become same, and another new strand produce, new double strand produce, then it is called conservative. But it did not follow this procedure. It follow the semi-conservative mode of DNA replication. Already they experimentally this proved in Maxwell style experiment. Right now we are not going in detail of that experiment. Here just you know that replication process is semi-conservative mode. Okay. How this occurs we'll discuss later on during the mechanism part. Okay. Now another one important point, that is origin of replication. Origin of replication. That means originate. From where the replication start. It called origin of replication. Okay. So this origin point also the part of DNA segment. It is not a different thing. It is also a part of DNA segments. Some repetitive DNA segments are present. Okay, some repetitive DNA segments are present. Okay, and which can be recognized by some multimeric origin binding protein. That means the point from where replication start, it called as origin of replication. Okay, from where it will start. Okay, so some identification marks are there. some repetitive DNA segments are present generally in that origin points, and that reasons that repetitive DNA segments can be recognized by some multimeric origin binding proteins. These multimeric origin binding proteins help other proteins or enzymes to initiate DNA replication. That means, suppose this multimeric origin binding proteins help to identify the region where replication starts for an enzyme like DNA polymerase, because DNA polymerase is one of the enzymes which is involved in this replication process, and another enzymes which are responsible for this replication processes. Okay. Another important point of this origin point, generally the point from where this replication starts, generally these regions are AT rich region. AT rich. Why the origin points are AT rich region? Because in my semi-conservative mode of DNA replication, I show that based on old DNA strand, these new DNA strands are produced. That means, already you know, two DNA strands are joined with the help of hydrogen bonds. Okay, so that means these hydrogen bonds should be break down so that it becomes single-stranded or new DNA strands synthesis occur. Okay, and for that, all of you know, if one strand containing adenine and its complementary nucleotide will be thymine, or if it containing guanine, it containing cytosine, other strand. Position is 5 to 3, polarity position is 3 to 5, or vice versa. Suppose if it containing cytosine, that it should be containing guanine, or if it contains thymine or sorry, like adenine, like that. Okay, so that means this adenine and thymine, these two nucleotides are joined with the help of these hydrogen bonds. So that hydrogen bonds should be break down so they become single-stranded. Okay? So, why AT rich region? Because between adenine and thymine, two hydrogen bonds are there. These are the hydrogen bonds. But between guanine and cytosine, 3 hydrogen bonds are there. So, it is more easy to break 2 hydrogen bonds than in comparison to 3 hydrogen bonds. That's why origin of replication, this region, these are generally rich in AT. So it is enzymatically more helpful to break these 2 hydrogen bonds in comparison to the containing nucleotides having 3 hydrogen bonds like GC base pairs. Okay, so this origin point normally containing rich of AT rich region. Okay, up to here we studied what is replication. So what is replication? Replication means it will increase the copy number. Copy number of what? Copy number of DNA, because we are studying here DNA replication. So how do you increase the copy number? Based on a parental DNA strand, because DNA are double-stranded molecules. So based on DNA double-stranded molecules, it will produce 2 daughter DNA molecules. During that synthesis process, from a parental, it will produce 2 daughter molecules. During that process, it will follow the semi-conservative mode of DNA replication. So what is semi-conservative mode of DNA replication? That means one strand is old. Based on that strand, new strand is synthesized. Like that, another, based on another old strand, new strand is synthesized. So that process is called as semi-conservative mode of DNA replication. Okay, and that replication process starts in some points, and that points are called origin of replication. Okay, and there are some speciality of these regions. Generally, this origin of replication regions, these have some repetitive segments, and these repetitive segments can be recognized by some multimeric origin binding proteins, and these proteins show the way, that means show the point where other proteins or enzymes which are involved in this process can bind. Like when this multimeric, the origin binding proteins bind with these DNA regions, then it will show other proteins like DNA polymers or other enzymes which are involved in replication process. Okay, and another important point of this origin of replication, that region generally rich in AT, because AT rich regions are easy to break the hydrogen bonds. Okay, now after that we are going to discuss the proteins or enzymes which are involved in that replication process.